You have learned inheritance pattern, genetic basis of such pattern. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of putative genetic material was investigated, culminating in the realization that DNA deoxyribonucleic acid is the genetic material, and at least for the majority of organisms. In class 11th, you have learned that nucleic acid are polymers of nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are two types of nucleic acid found in living system. DNA acts as the genetic material in most of the organism. RNA through it also acts as a genetic material in some viruses. Mostly functions are messengers. RNA has additional roles as well. It functions as adapter structure and in some cases catalytic molecule. In class 11th, you have already learned the structure of nucleotides and the way these monomers units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from the DNA transcription, the genetic code that determines the sequence of amino acid in protein, the process of protein synthesis, translation, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set a new era of genomics. In the last section, the essential of human genome sequencing and its consequences will also discussed. Let's us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent section, we will understand that why it is most abundant genetic material and what its relationship is with RNA. 5.1 The DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as a number of nucleotides or a pair of nucleotides referred to as base pair present in it. And this also is the characteristic of an organism. For example, a bacteriophage has known as 5 into 174 has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pair, and haploid content of human DNA 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such long polymer. 5.1.1 Structure of polynucleotide chain. Let us recapitulate the chemical structure of polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose in case of DNA, and a phosphate group. There are two types of nitrogenous bases. First, purine, and this contain adenine and guanine, and second, pyrimidines, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA. Thymine is present in DNA. Thymine present in DNA and uracil present in RNA. At the place of thymine, a nitrogenous base is linked to OH of 1 carbon pentose sugar through N-glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cysteine or deoxycysteine, and uridine or deoxythymidine. When a phosphate group is linked to OH of 5 carbon of a nucleoside through phosphate linkage, a corresponding nucleotide or deoxyribonucleotide, depending upon the type of sugar present, is formed. Two nucleotides are linked through 3-5' phosphodiester linkage to form a dinucleotide. Two nucleotides linked with phosphodiester linkage. More nucleotides can join in such a manner to form a polynucleotide chain. A polymer thus formed has at one end free phosphate moiety 5' end of sugar, which referred to as 5' end of polynucleotide chain. Similarly, at the other end of polymer, the sugar has a free OH or 3' carbon group, which is referred to as 3' end of polynucleotide chain. The backbone of polynucleotide chain is formed due to sugar and phosphate. The nitrogenous base linked to sugar moiety project from backbone of polynucleotide chain. 5' phosphate, 3' hydroxy group, and these nucleotides are joined by phosphodiester linkage. In RNA, every nucleotide residue has an additional OH group present at 2' position in ribose. Also, in RNA, uracil is found at the place of thymine. Thymine, also known as 5-methyl-uracil, another chemical name of thymine. DNA is an acidic substance present in nucleus, was first identified by Friedrich Miescher in 1869. He named it nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chain. However, this proposition was also based on the observation of Irving Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant. This is known as Chargaff rule. Ratio of these A and T and G and C are constant. The base pairing confirms a very unique property of polynucleotide chain. They are said to complementary to each other, and therefore then there is a sequence based on strand of DNA. Then sequence is other strand that can be predicted. Also, if each strand of DNA acts as template for synthesis of new strand, the double-stranded DNA thus produce would identical to parental DNA molecule. Because of this, genetic implication of structure of DNA become very clear. The silent feature of double helix structure of DNA. First, it is made up of two polynucleotide chain, where the backbone is constituted by sugar phosphate and base pair projected inside. Second, the two chains have antiparallel polarity. It means if one chain has polarity 5' to 3', the other has 3' to 5'. The base in two strands are paired through hydrogen bond, forming a base pair. Adenine forms two hydrogen bond with thymine. Adenine form two bond with thymine, and cytosine form guanine form three hydrogen bond with cytosine. Generate approximately uniform distance between strands of helix. 4. The two chains are coiled in right-handed fashion. The pitch of helix is 3.4 nanometer. 1 nanometer is billionth of meter, means 1 nanometer is equal to 10 to the power minus 9 meter, and there are roughly 10 base pair in each turn. Consequently, the distance between a base pair in a helix is 0.34 nanometer. 4. The plane of one base pair stacks over the other in double helix. This in addition of hydrogen bond confers the stability of helical structure. Double stranded polynucleotide chain, GNC, 3 here 3 hydrogen bond, here between A and T and 2 hydrogen bond. This nucleotide binds with this by phosphodiester linkage. And next, adenine thymine base pair, backbone, sugar phosphate backbone, and this is base pair. Compare the structure of purine and pyrimidine. Can you find out why the distance between the two polynucleotide chain in the DNA remain almost constant? Because one is adenine, one is thymine. प्यूरिन एक है और एक पैरिमिडीन है, डेट्स वाई डीएनए हेलिक्स, डबल डीएनए हेलिक्स गिवन बाय फ्रांसिस एंड क्रिक इन 1957. किस ईयर में दिया गया भाई? 1953, सॉरी. Yeah, 2 dates. The proposition of double helix structure of DNA and simplicity in explaining genetic implication become revolutionary. Very soon, Francis Crick proposed central dogma molecular basis, which stated that genetic information flow from DNA to RNA to protein. This is given by Francis Crick, also known as central dogma. DNA replication हो रहा है, फॉर्म करेगा डीएनए, ट्रांसक्रिप्शन आरएनए, और ट्रांसलेशन होगा तो प्रोटीन बना रहा है. इसको क्या बोलेंगे? सेंट्रल डोग्रा. दिया कौन? क्रिक. In some viruses, the flow of information is reverse direction, that is from RNA to DNA. This is known as reverse transcription. You can see in ads, the virus can convert DNA from RNA. That is known as reverse transcription. 5.1.2 Packaging of DNA helix. Taken the distance between two consecutive base pairs is 0.34 nanometer, 0.34 into 10 to the power minus 9 meter. If the length of DNA double helix in typical mammalian cell calculated simply by multiplying the total number of base pair with distance between two consecutive base pairs. 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power 9 base pair, it come out to be approximately 2.2 meters. A length that is far greater than the dimension of typical nucleus, approximately 10 to the power minus 6 meter. How is such long polymer packaged in cell? Nucleus. Here is core of histone molecules, DNA, H1 histone protein, histone octamer. If the length of Escherichia coli DNA is 1.36 mm, can you calculate the number of base pair in Escherichia coli? Number of base pair, then we can divide 1.36 mm by 0.34 by calculating this into 10 to the power minus 9 और इसको सॉरी क्या है भाई 1.36 into 10 to the power minus 3 by 0.34 into 10 to the power minus 9. We can get number of base pairs in Escherichia coli by dividing this. This is one way divided by 4, 4, 5, approx 3.6 into 10 power 6. 3 point something into 10 power 6. In prokaryotes such as Escherichia coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged, DNA is negatively charged, is held with some protein that have positive charge, held by some protein that are positive charge in a region termed a nucleoid. The DNA in nucleotide is organized in large loops held by proteins. Electron microscope picture of bits on a string. In eukaryotes, this organization is much more complex. There is a set of positive charged basic protein called histones. A protein acquires charge depending upon the abundance of amino acid residue with charged side chain. Histones are rich in basic amino acid residues, lysine and arginine. Both the amino acid residues carry positive charge in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. Thread-like stand color bodies seen in nucleus. The nucleosome in chromatin are seen as bits on a string structure when viewed under electron microscope EM. Theoretically, how many such bits nucleosome do you imagine are present in mammalian cell? The bits on a string structure in chromatin is packed to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packing of chromatin at higher level require additional set of protein that collectively are referred to as non-histone chromosomal protein. The packaging of chromatin at higher level requires additional set of protein referred to as non-histone chromosomal protein. In a typical nucleus, some region of chromatin are loosely packed and stand light and are referred to as euchromatin. The chromatin that is more densely packed and stand dark are called heterochromatin. Heterochromatin densely packed and stand dark. Euchromatin is said to be transcriptionally active to form active chromatin where heterochromatin is inactive. 5.2 The search for genetic material. Even through the discovery of nuclein by Miescher and the proposition of principle of inheritance by Mendel were almost at the same time, but that the DNA acts as genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism bacteria had changed a physical form. When Streptococcus pneumococcus bacteria are grown on culture plate, some produce smooth, shiny colonies, while others produce rough colonies. This is because the S strain bacteria have a mucus polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with R strain do not develop pneumonia. Okay. Virulent die. S strain injected in mice die. R strain injected in mice live. Griffith was able to kill bacteria by heating them. He observed that heat killed the S strain bacteria injected in mice did not kill them. When he injected a mixture of heat killed S strain and live R strain, the mice died. Moreover, he recovered living S bacteria from dead mice. He concluded that the R strain bacteria had somehow been transformed by heat-killed S strain bacteria. Some transforming principle transferred from heat-killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Here is Griffith experiment. He performed in 1928 on pneumonia bacteria on mice. Okay, S strain mice killed, R strain mice live. Heat killed S strain mice live. Heat killed S strain plus R strain live, mice dead. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, MacLeod and McCarthy in 1934 to 1944, 10 years, the genetic material was thought to be protein. They worked to determine the biochemical nature of transforming principle in Griffith experiment. They purified biochemicals, that is protein, DNA, RNA, etc., from the heat-killed S cell to see which one could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzyme protease and RNA digesting enzyme RNase did not affect transformation, so the transforming substance was not protein or RNA. Digestion with DNA did inhibit the transformation, suggesting that DNA caused the transformation. They concluded that DNA is hereditary material, but not all biologists were convinced by transformation principle given by Avery, MacLeod and McCarthy. 5.2.1 The genetic material is DNA. The unequivocal proof that DNA is genetic material came from the experiment of Alfred Hershey and Martha Chase in 1952. Unequivocal proof of DNA is genetic material given by Hershey and Martha Chase in 1952. Okay. They worked with viruses that infect bacteria called bacteriophage. Bacteriophage infect viruses. The bacteriophage attached to. They worked with viruses that infect bacteria called bacteriophage. Bacteriophage infect bacteria. These are type of viruses. The bacteriophage attached to bacteria and its genetic material enter in bacterial cell. The bacterial cell treat the viral genetic material as if it was its own and subsequently manufactures more viruses particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. They gave some viruses on the medium that contain radioactive phosphorus and some other medium that contain radioactive sulfur. Viruses grown in presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains the phosphorus but protein does not. Similarly, the viruses grow on radioactive sulfur containing radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli. Then as infection proceeds, the viral Coats were removed from the bacteria by agitating them in blender. The virus particles were separated from the bacteria by spinning them with centrifuge. Bacteria which was infected with virus that has radioactive DNA were radioactive, indicating that DNA was material that passed from virus to bacteria. Bacteria that were infected with virus that had radioactive protein were not radioactive. This indicates that protein did not enter the bacteria from virus. DNA is referred to genetic material that is passed from viruses to bacteria. Here, Hershey and Chase experiment. Bacteriophage virus, radioactive 335 sulfur, radioactive phosphorus P32. Okay, infection first stage, second stage blending, third centrifugation. Radioactive phosphorus detected in cell, no radioactive sulfur detected in cell. This indicates the genetic material contain phosphorus. Radioactive phosphorus means transforming principle. The component caused transformation is DNA. Radioactive sulfur 35 detect the supernatant. No radioactive detected in supernatant. 5.2.2 Property of genetic material DNA versus RNA. From the foregoing discussion, it is clear that debate between proteins versus DNA as genetic material was unequivocal result from Hershey-Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it is subsequently become clear that in some viruses, RNA is genetic material. For example, tobacco mosaic virus, QB bacteriophage, etc. Answer to some questions such as why DNA is predominant genetic material, whereas RNA performs dynamic function of messenger and adapter has to be found from the difference between chemical structure of two nucleic acid. Can you recall two chemical differences between RNA, DNA and RNA? A molecule that can act as genetic material must fulfilling the following criteria. First, it should be able to generate its replication. Second, it should be stable chemically and structurally. Third, it should provide a scope for slow chain mutation that required for evolution. Fourth, it should be able to express itself from Mendelian character. If one examines each requirement on one by one, because of rule of basic pairing and complementarity, both the nucleic acid DNA and RNA have the ability to direct their duplication. The other molecule in living systems such as protein fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the property of genetic material. This now can easily be explained in light of DNA that the two strands of being complementary if separated by heating come together when appropriate conditions are provided. Further, 2' OH group present at every nucleotide in the RNA is a reactive group and marks RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among two nucleic acids, DNA is better genetic material. DNA is better genetic material. Okay. In fact, the presence of thymine at the place of uracil, thymine present in DNA and uracil present in RNA. Also conformational stability of DNA. 5' uracil, also known as detailed discussion about required understanding of process and development of DNA, and you will study this in process in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at the faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for synthesis of protein, hence can easily express the characters. DNA, however, is independent on RNA for synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicates that both RNA and DNA function as genetic material, but DNA being more stable preferred for storage of genetic information. For transmission of genetic information, RNA is better. For transmission of genetic information, RNA is better, but for storage of genetic information, RNA is preferred. RNA word 5.3. From foregoing discussion, an immediate question become evident: which is first genetic material? It shall be discussed in detail in the chapter Chemical Evolution, but briefly we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life process such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that make it more stable. DNA being double-stranded and having complementary strand further changed by evolving process of repair. Replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme of replication of DNA to quit their original statement that is as follows. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for genetic material. Watson and Crick in 1953. The scheme suggested that the two strands would separate and act as template for synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Watson and Crick model for semi-conservative DNA replication. 5.4.1 Experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plant and human cells. Matthew Michelson Franklin Stahl performed the experiment in 1953. Semi-conservative nature of DNA of replication given experimentally shown by Michelson and Stahl in 1958. If they grew Escherichia coli, a medium containing NH4Cl, nitrogen is heavy isotope of nitrogen. This nitrogen is heavy isotope of nitrogen. Is the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen-containing compounds. This heavy DNA molecule could be distinguished from normal DNA by centrifugation in cesium chloride. Density gradient. Please note that N15 is not a radioactive isotope. It is separated from N15 only based on density. It is separated on the basis of density. Second, then they transfer to the cell into medium with normal NH4Cl. This is normal ammonium salt, and took sample various different time interval as the cell multiplied and excreted the DNA that remained as double-stranded helices. The various samples were separated independently on cesium chloride gradient to measure the density of DNA. Can you recall what centrifugal force is and think why molecule with higher mass density would sediment faster? The results shown in figure 5.7 Michelson and Stahl experiments. Separation of DNA by centrifugation. Gravitational force N heavy, hybrid, light, hybrid. After one generation, semi-conservatively divide in हाफ डिवाइड हुआ, ठीक। इस मीडियम 14 के मीडियम में ग्रो कराया तो 2 इसका हाफ डिवाइड हुआ, फिर 20 मिनट के बाद एक जनरेशन कंप्लीट हुआ, फिर 40 मिनट बाद क्या 2 जनरेशन कंप्लीट हुआ। तो इन दोनों का सेमी कंजर्वेटिव रिप्लिकेशन हुआ, ठीक। तो एक इससे इनकॉर्पोरेट हो गया, एक इससे, ठीक। इसका दो फॉर्म बनेगा, एक यहाँ एक यहाँ, फिर एक जो मीडियम था उससे हो गया 2 हाइब्रिड and 2 pure. Thus, the DNA that was excreted from the culture one generation after the transfer of N15 to N40 medium, that is after 20 minutes, is circle divided in 20 minutes, had a hybrid of intermediate density. DNA extracted from culture after another generation, that is 40 minutes. Second generation was composed of equal amount of hybrid DNA and light DNA. Okay. If the cell cycle is allowed to grow for 80 minutes, then what would be proportion of light and hybrid DNA? This 80 मिनट देखो, प्योर, अरे भाई बनाना पड़े, 80 मिनट का बात कर रहा ना, तो 40 मिनट में हुआ, फिर 60 मिनट देख लो, 80 मिनट 60 मिनट में क्या हुआ, इसका 2 इधर से 2 प्योर, यहाँ से 2 प्योर, यहाँ से 2 प्योर, यहाँ से 2 प्योर, 2 हाइब्रिड, मतलब कि 4 8 प्योर, 4 हाइब्रिड, फिर यहाँ से यहाँ देखो तो क्या हो गया, 16 हाइब्रिड एंड 16 प्योर 8 हाइब्रिड अप्रॉक्स होना चाहिए। सॉल्व करना पड़ेगा, रुक जाओ। वेरी सिमिलर एक्सपेरिमेंट इन्वॉल्विंग यूज ऑफ रेडियोएक्टिव थायमिडीन, दिस इज वेरी इंपॉर्टेंट। Very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on VCF fava beans by Taylor and colleagues in 1958. The experiment proved that DNA in chromosomes also replicate semi-conservatively. 5.4.2 The machinery of and the enzymes. In living cells such as Escherichia coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in very short time. Escherichia coli that has only 4.6 and 10.6 base pair compare it with human. Those deployed content is 6.6 and 10.9 base pair. Complete the process of replication within 18 minutes. That means the average rate of polymerization has been approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they have also to catalyze the reaction with a very high degree of accuracy. Any mistake during the replication would result into mutation. Furthermore, energetically replication is a very expensive process. Deoxyribonucleoside triphosphate serve dual purpose. In addition to acting as substrate, they provide energy for polymerization reaction. The two-terminal phosphate in deoxyribonucleoside triphosphate are highly energy phosphate, same as in case of ATP. In addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to high energy requirement, the replication occurs within a small opening of DNA helix referred to as replication fork. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3'. This creates some additional complication at the replicating fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous, while on other, the template with polarity 5' to 3', it is discontinuous. The discontinuously synthesized fragments are later joined by enzyme DNA ligase. The DNA polymerase on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is definite region Escherichia coli DNA where the replication originates. Such region is known as origin of replication. It is because of requirement of their origin of replication that a piece of DNA, if needed to be propagated during the recombination of DNA procedures, require a vector. The vector provides an origin of replication. Further, not every detail of replication is understood well. In eukaryote, the replication of DNA takes place at S phase of cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, a chromosomal anomaly. You will learn detailed nature of origin and process occurring at this site in higher classes. Replicating for continuous synthesis, template DNA or parental synthesis as parental strand, discontinuous synthesis, and in these are known as Okazaki fragments, and these are synthesized in 5' to 3' direction, and in continuous, and these are synthesized in 5' to 3'. Okay, discontinuous 5' to 3' synthesis, and continuous 5' to 3' synthesis. 5.5 transcription. The process of copying genetic material information from strand of DNA into RNA is termed transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now form base pair with uracil instead of thymine. Now, however, unlike the process of replication, which once set in, the total DNA of an organism get duplicated. In transcription, only a segment of DNA and only one strand is copied into RNA. This necessitates defining the boundary that would demarcate the region and strand of DNA that would transcribe. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as template, they would code for DNA molecule with different sequence. Remember, complementarity does not mean identical, and in turn, if they code for protein, the sequence of amino acid in protein would be different. Hence, one segment of DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein and exercise. The transcription would become futile one. 5.5.1 transcription unit. Our transcription unit in DNA is defined primarily by three regions in DNA. First, promoter, second, the structural gene, third, terminator. There is convention in defining the two strands of DNA in the structural gene and transcription unit. Since two strands have opposite polarity and DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3'. Two strands of opposite polarity DNA-dependent RNA polymerase also catalyze the polymerization in 5' to 3'. The strand that has polarity 3' to 5' acts template and is also referred as template strand. The other strand that has polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil is displaced during the transcription. Strangely, this strand which does not code for anything is referred to as coding strand. All the reference point while defining a transcription unit is made of coding strand. To explain the point or hypothetical sequence from transcription unit is represented below. Coding strand, template strand. Schematic structure of transcription unit: 3' to 5', template strand, coding strand, 5' to 3', and this is structural gene. Here is structural gene. This is template strand. Here is promoter. Transcription start from promoter and end at terminator. The promoter and terminator flank structural gene in a transcription unit. The promoter is said to be located toward 5' end upstream of structural gene. The reference is made with respect to polarity of coding strand. It is DNA sequence that is provided by binding site for RNA polymerase, and it is presence of promoter in a transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strand could be reversed. The terminator is located toward 3' end downstream of the coding strand, and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of this sequence shall be discussed while dealing with regulation for gene expression. 5.5.2 transcription unit and the gene. A gene is defined as a functional unit of inheritance through there is no ambiguity that genes are located in DNA. It is difficult to literally define the gene in terms of RNA sequence. The DNA sequence coding for tRNA or RNA molecule also defined as gene. However, by defining cistron as a segment of DNA coding for polypeptides, the structure gene in a transcription unit could be said to be monocistronic, mostly in eukaryote, polycistronic, mostly bacteria and prokaryote. Okay. In eukaryote, the monocistronic structural gene have interpreted coding sequence. The gene in eukaryote are split. The coding sequences are expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns are intervening sequences do not appear in mature RNA or proceed RNA. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. Inheritance of character is also affected by promoter and regulatory sequence of a structure gene. Hence, sometimes the regulatory sequence are loosely defined as regulatory gene, even though these sequences do not code for any RNA or protein. 5.5.3 Type of RNA and process of transcription. In bacteria, there are three major types: RNAs, mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNA are needed for synthesis of protein in cell. The mRNA, messenger RNA provide the template, tRNA bring amino acid and treat the genetic code, and rRNA play a structural and catalytic role in translation. There is a single DNA-dependent RNA polymer that catalyzes the transcription of all type of RNA in bacteria. RNA polymerase bind to promoter and initiate transcription, that is initiation. It used to nucleoside triphosphate H phosphate and polymerize the template dependent fashion follows the rule of complementarity. It somehow also facilitates the opening of helix and continuous elongation. Only a short stage RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also RNA polymerase. This results in termination of transcription. Here is process of transcription in bacteria. Initiation. Promoter is rho sigma factor. This is RNA polymerase. DNA helix. Elongation. Termination. Here is elongation. Here is rho factor comes at termination point. An intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which initiation, elongation and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiator factor sigma and termination factor rho to initiate and terminate the transcription respectively. Initiation done by sigma factor and termination done by rho factor. Association with these factors alter specificity of the RNA polymerase to either initiate or terminate. In bacteria, since mRNA does not require any process to become active and also transcription and translation take place in same compartment, there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. First, there are at least 3 RNA polymerase in nucleus. In addition to RNA polymerase found in organelles, there is a clear-cut division of labor. The RNA polymerase first transcribe RNAs 28S, 18S and 5.8S, whereas RNA polymerase thought responsible for transcription of tRNA and 5S RNA and S and RNA. Small nuclear RNA. The RNA polymerase second transcribe the precursor of mRNA, the heterogeneous nuclear RNA, HNRNA. The second complexity is that primary transcripts contain both exons and introns and non-functional. Hence, it is subjected to the process called splicing, where introns are removed and exons are joined in a defined order. HNRNA undergoes addition process called capping and tailing. as an RNA undergoes capping and tanning. In capping, an unusual nucleotide methylguanidine triphosphate is added to 5 ends of hnRNA. In tanning, adenylate residue 200 to 300 are added to 3' end in template independent manner. It is fully processed hnRNA, now called mRNA, that is transported out of the nucleus of translation. The significance of such complexity is now beginning to understand. The split gene arrangement represents probably an ancient feature of genome. The presence of introns is reminiscent of antiquity, and process of splicing represent the dominance of RNA work. In recent times, the understanding of RNA and RNA-dependent processes in giving system have assumed more importance. 5.6 genetic code. During the replication, transcription of nucleic acid was copied to form another nucleic acid. Hence, these processes are easily conceptualized and on the basis of complementarity. The process of translation required transfer of genetic information from a polymer of nucleotide to synthesize a polymer of amino acid. Neither does any complementarity exist between nucleotides and amino acid, nor could any be drawn theoretically. There exists ample evidence to support the notion that change in nucleic acid genetic material were responsible for change in amino acid in proteins. This led to proposition of genetic code that could direct the sequence of amino acid during the synthesis of protein. If determining the biochemical nature of genetic material and the structure of DNA was very exciting in proposition, in deciphering of genetic code were most challenging. In very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, geneticists, etc. George Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should be constituted a combination of bases. He suggested that in order to code for 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because permutation combination 4 into 4 would generate 64 codons. Generating many more codons are required, providing the proof that codon was triplet was more daunting task. The chemical method developed by Har Gobind Khurana, instrumental in synthesizing RNA molecule, defines the combination of bases, homopolymer and copolymer. Marshall Nirenberg cell-free system for protein synthesis finally helped the code for deciphered. Severo-Okai enzyme, polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequence in template-independent manner. Enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in figure 5.1. The codons for various amino acids. First position, third position, second position. Here is UA important, tyrosine, UAC, tyrosine, UAA stop, UAG stop, UGA stop codon, AUG methionine, AA lysine, AAG lysine. Silent feature of genetic code. The code is triplet, 61 codons code for amino acid and 3 codes for 3 not code amino acid stop codon. AUG, what? UAA, UAG and UGA stop codon. Some amino acids are coded by more than one codon, hence code is degenerate. Third, the codon is read in mRNA in a contiguous fashion. There are no punctuation. Fourth, The code is nearly universal. For example, from bacteria to human, triple U would code phenylalanine. Some exceptions to this rule have been found in mitochondrial codon and in some protozoans. AUG has dual nature, is code for methionine and is also initiator codon. The UAA, UAG, UGR stop terminating codon. If following is sequence of nucleotide mRNA predict the sequence of amino is coded for the following: AUG start codon, UUU code for phenylalanine, UUC here UUC phenylalanine and phenylalanine और ये भी वही होगा, ये भी वही UEC. Now try to oppose following the sequence of code मेथोनिन phenylalanine. ये तो इम्पोर्टेंट है भाई, याद करना पड़ेगा. phenylalanine का UUC 5.6.1 mutation and genetic code. The relationship between gene and DNA are best understood by mutation and studies. You have studied about mutation and its effect in chapter 4. Effects of larger deletion and rearrangement in segment of DNA are easy to comprehend. It may result in loss or gain of gene and so as function. The effect of point of mutation will be explained here. A classical example of point mutation in change in single base pair in gene for beta globin chain that result in change of amino acid residue glutamate to olein. It results in disease called sickle cell and anemia. Effect of point mutation that insert or delete of base of structure gene can be better understood by following simple example. Consider the statement that made by following each word having 3 letter genetic code. B has rearranged statement itna. Ram has B. Similarly, new should BI. Now it should be read B. Now the conclusion from the above exercise is insertion or deletion of one or two bases change the reading frame from the point of insertion or deletion. However, such mutation is referred to as frame shift insertion or deletion. Insertion or deletion of 3 or its multiple bases insert or delete in one or multiple codon. Hence, one multiply amino acid and reading frame require unaltered from that point onward. 5.6.2 tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be mechanism to read the code and also the link to amino acids, because amino acids have no structural specialties to read the code unequally. They postulate the presence of an adapter molecule that on one hand read the code and on other hand would bind to the specific amino acid. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor into which it binds to amino acid. tRNA is specific for each amino acid. For example, for to initiation there is another specific tRNA that is referred to initiator tRNA. There are no tRNA for stop codon. In figure 5.12, the secondary structure of tRNA has been depicted that look like clover leaf. In actual structure, the tRNA compact molecule which look like inverted L. tRNA adapter molecule, serine, tyrosine, tRNA 5' and 3' UCA, AUGU, anticodon, codon, tRNA 5' and 3' AUG, UAC, mRNA 5' to 3'. 5.7 Translation. Translation refers to the process of polymerization of amino acid to form polypeptide. The order of sequence of amino acids are defined by sequence of bases in mRNA. The amino acids are joined by bond which is known as peptide bond. Formation of a peptide bond require energy. Therefore, in first phase itself, amino acids are activated in presence of ATP and linked to their cognate tRNA. A process commonly called charging of tRNA or amino acylation of tRNA to be more specific. If two such charges tRNA are brought close to enough, the formation of peptide bond between them would be favored energetically. The presence of catalyst would enhance the rate of peptide bond formation. Translation. The cellular factory responsible for synthesizing a protein is ribosome. The ribosome consists of structural RNA and about 80 different proteins. In its inactivate state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. There are two sites in large subunit for subsequent amino acid to bind to and thus to be close enough to each other for the formation of a peptide bond. The ribosomes also act as catalyst 20S rRNA in bacteria in enzyme ribozyme for formation of peptide bond. A translational unit in mRNA is the sequence of RNA that is flanked by start codon AUG and in the stop codon and the codon for polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated region UTR. The untranslated region are present at both 5' ends start before start codon and 3' ends after stop codon. They are required for efficient translation process. For initiation, the ribosome bind to mRNA and start codon AUG that is recognized by the initiator RNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, the complex is composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming the complementary base pair with tRNA anticodon. The ribosomes move from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequence dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from ribosome. 5.8 Regulation of gene expression. Regulation of gene expression refers to a very broad term that may not occur at various levels. Considering that gene expression results in the formation of polypeptide, it can be regulated at several levels in eukaryotes. The regulation could be executed as transcription level, formation of primary transcript; second, processing level, regulation of splicing; third, transport of mRNA from nucleus to the cytoplasm; fourth, translational level. The gene in the cell expressed to perform particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by Escherichia coli, it is used to catalyze the hydrolysis of disaccharide lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to utilized energy, they would no longer require the synthesis of enzyme beta-galactosidase. Therefore, in simple terms, it is metabolic, physiological, and environmental conditions that regulate the expression of gene. The development and differentiation of embryo into adult organism and also result in coordinated regulation of expression of several set of genes. In prokaryotes, control the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoters is in turn regulated by interaction with accessory protein, which affect the ability to recognize start site. These regulatory proteins can act both positively activators and negatively repressors. The accessibility to promoter region of prokaryotic DNA is in many cases regulated by interaction of proteins with sequence termed as the operator. The operator region is adjacent to the promoter element in most operons, and in most cases the sequence of the operator bind a repressor protein. Each operon has its specific operator and a specific repressor. For example, like operator is present only in the like operon, and it interacts specifically with like repressor only. 5.8.1 like operon. The elucidation of like operon was also a result of close association between geneticist Francois Jacob and biochemist Jacob Monod. They were first to elucidate the transcriptionally regulated system. In like operon, here like refers to lactose or polycystonic structure gene is regulated by a common promoter and regulatory gene. Such arrangement is very common in bacteria and referred to as operon. To name few such examples, like operon, TRP operon, ARA operon, His operon, Valine operon, etc. Like operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from word inhibitor. And 3 structure gene ZYA. The I gene codes for repressor of like operon. The Z gene codes for beta glycosidase, beta gal, which is primarily responsible for hydrolysis of disaccharide lactose into monomeric unit galactose and glucose. The Y gene codes for the permease, which is increasing permeability of cell to beta galactosidase. The A gene encodes the transacetylase. Hence, all 3 gene products in like operon are required for metabolism of lactose. In most other operon as well, the genes present in operon are needed to function in same or related metabolic pathway. Like operon, in absence of inducer, repressor bind to operator. Repressor bind to operator region prevent the RNA polymerase to transcribe the operon. In inducer, in presence of inducer bind to repressor, then Z form beta glycosidase permease, Y gene permease, A gene by translation transacetylase. Lactose is a substrate for enzyme beta galactosidase and it regulates the switching on and off of operon, hence it determines inducer. In the absence of preferred carbon source such as glucose, if lactose is provided with growth medium of bacteria, the lactose is transported into cell through action of permease. Remember, a very low level of expression of like operon has to be present in cell all the time, otherwise lactose cannot enter the cell. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to operator region of the operon and prevents the RNA polymerase from transcribing the operon. In the presence of inducers such as lactose, allolactose, the repressor is inactivated by interaction with inducer. This allows the RNA polymerase access to promoter and transcription proceed. Essentially, the regulation of lac operon can also be visualized as regulation of enzyme synthesis by substrate. Remember, glucose or galactose cannot act as inducer for lac operon. Can you think how long lac operon would express in the presence of lactose? Regulation of lac operon is by repressor reproduce negative regulation. Lac operon is also positive regulation, but it is beyond our scope. 5.9 Human Genome Project. In the preceding section, you have learned the sequence of basic DNA determines the genetic makeup of a given organism. In other words, genetic makeup of organism or individual lie in the DNA sequence. If two individuals differ, their DNA sequence should also be different, at least at some places. This also led to the quest of finding out complete DNA sequence of human genome with the Australian genetic engineering technique, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques of determining DNA sequence. A very ambitious project of human sequence launched in the year 1990. Human Genome Project (HGP) was mega project. You can imagine the magnitude and requirement of project if we simply define the aim of project follows. Human genome is said to be approximately 309 base pair, and if cost of sequencing about $3 per base pair, estimated cost to be $9 billion. Further, if the obtained sequence were stored in type of form of book, and if each book contained 1,000 letters and each book contained 1,000 pages, and there are 33,300 such books would be required to restore the information of DNA sequence in single human cell. The enormous amount of data expected to generate also necessitated to use high-speed computational devices for the storage and retrieval and analysis. Human Genome Project was closely associated with the rapid development of new area of biology known as bioinformatics. Goals of Human Genome Project. Some of the important goals of Human Genome Project: first, identify all of approximately 27,000-25,000 genes in human DNA. Determine the sequence of 3 billion chemical base pairs that make up the human DNA. Third, store this information in database. 4, improve tool for data analysis. Fifth, transfer related technology to other sectors such as industry. 4, address the ethical, legal and social issue that may arise from the project. The Human Genome Project, a 30-year-old project coordinated by U.S. Department of Energy and National Institute of Health. During the early year of Human Genome Project, the Wellcome Trust UK became the major partner. Additional contribution came from Japan, France and Germany, China and others. The project was completed in 2003. Knowledge about the effects of DNA variation among individuals can lead to the replacement new ways of diagnosis. Treat someday, prevent the thousands of disorders that affect the human being. Besides the providing clue that understanding human biology, learning about non-human organism DNA sequence can lead to the understanding of the natural capability that can be applied towards the solving challenges, healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Cynorhabditis elegans, a free-living non-pathogenic nematode, Drosophila, fruit fly, plant rice and Arabidopsis, etc. have also been sequenced. Methodologies. The method involved in two major approaches. One approach focused on identifying the genes that express as RNA, referred to express sequence tag EST. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence, the latter assigning different regions in the sequence with function and term as sequence annotation. For sequencing the total DNA from the cells isolated and converted into random fragment and collectively smaller size of recall is very long polymer, and there is technical limitation in sequence very long piece of DNA and clone in suitable host using specialized vector. The cloning results in amplification of each piece of DNA fragment. so that it subsequently could sequence with each and commonly used host where bacteria and yeast, and the vectors were called BAC, bacterial artificial chromosomes, and YAC, yeast artificial chromosomes. The fragments were sequenced using automated DNA sequencer that works in principle method developed by Frederick Sanger. Remember, Sanger is also credited for developing method of determination of amino acid sequence protein. These sequences were arranged in bases some overlapping region present in them. This required generation and overlapping of fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosomes. The sequence of chromosomes 1 was completed in 2006. This was last and of 24 chromosomes, 22 autosomes and XY to be sequenced. Here representative diagram of Human Genome Project. Another challenging task was assigned to genetic and physiological map of human genome. This was generated using information of polymorphism and restriction of endonuclease recognition site and some repetitive DNA sequence known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be explained in next section of DNA fingerprinting. 5.9.1 Salient features of human genome. Some of the salient observations drawn in Genome Project is for the first, human genome contains 3,164.7 billion base pairs. Second, the average gene consists of 3,000 bases, but size vary greatly with largest human non-gene dystrophin 2.4 million bases. Third, the total number of genes estimated to be 30,000, much lower than the previous estimated 80,000 to 140,000 genes. Almost all 99.9% nucleotide bases exactly same in all peoples. 4. Functions are unknown over 50% of discovered genes. 5. Less than 2% of genome codes for protein. 6. Repeated sequence make a very large portion of human genome. 7. Repetitive sequence of stretched DNA that are repeated over time, sometimes 100 or 1000 times. They are through not direct coding function, but they shed light on chromosome structure, dynamics and evolution. 8. Chromosome first has most genes, 2968, and the Y has fewest, 231. 9. Scientists have identified about 1.4 million locations where single base DNA differences, SNP, single nucleotide polymorphism, pronounced to be SNPs, occur in humans. This information promises to revolutionize the process of finding chromosomal location for disease-associated sequence and tracing human history. 5.9.2 Application and future challenges. Deriving the meaningful knowledge from DNA sequence will define research through the coming decade, leading to our understanding of biological system. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sector worldwide. One of the greatest impact of having human genome sequence may be enabling the radically new approach to biological research. In the past, researchers studied one or few genes at a time with whole genome sequences and new height throughout technology. We can approach questions systematically and on a much broader scale. They can study all the genes in the genome, for example, all transcripts in particular tissue of organ and tumor, or how tens or thousand genes proteins work together in interconnected network or orchestra in chemistry of life. 5.10 DNA fingerprinting is studied in preceding section. 99.9% of base sequences among humans is same, assuming human genome as 3 into 10 to the 9 base pair. In in how many base sequences would there be difference? It is these differences in sequence of DNA which make very individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare two sets 3 into 10 power 6 base pairs. DNA fingerprinting is a very quick way to compare DNA sequencing in two individuals. DNA fingerprinting involves identifying the differences of some specific region in DNA sequence called repetitive DNA. Because in this sequence a small strand DNA is repeated over time, these repetitive DNA are separated from bulk genomic DNA as genetic peak during density gradient and centrifugation. The bulk DNA forms the major peak and the other small peaks are referred to as satellite DNA. Depending on the basis composition, A, T, rich and GC rich, length of segment or number of repetitive units, the satellite DNA is classified into many categories, such as microsatellite and mini satellite. These sequences normally do not code for any protein, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basic DNA fingerprinting. Since DNA from every tissue such as blood, hair follicles, skin, bone, saliva is formed, etc., from individuals show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphism are inheritable from parent to children, DNA fingerprinting is the basic of paternity testing in case of disputes. As polymorphism in DNA sequence, the basic of genetic mapping, the human genome as well as DNA fingerprinting, it is essential that we understood what DNA polymorphism means in simple terms. Polymorphism varies at genetic level, arises due to mutation, different kind of mutation and their effect that you have already studied in chapter 4 and in case of preceding section in this chapter. New mutation may arise in individual either in somatic cell or in the germ cell. Cells that degenerate the sexually reproducing organism. If a gene cell mutation does not seriously impair individual ability to have offspring who cannot transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic, again recall the definition of alleles from chapter 4. Allelic sequence variation had traditionally been described as DNA polymorphism. If more than one variant allele at the locus occur in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in population at higher frequency, it is referred to as DNA polymorphism. The probability of such variation to observed in non-coding DNA sequence would be higher as mutation in this sequence may not have any immediate impact in an individual reproductive ability. These mutations keep the accumulating generation after generation and form one basic variability and polymorphism. There is variety of different type of polymorphism ranging from single nucleotide change to very large scale changes. For evolution and speciation, such polymorphism play a very important role, and you will study these in detail in higher classes. The technique of DNA fingerprinting was initiated developing by Alec Jeffrey. He used genetic DNA as probe that show very high degree of polymorphism. It was called variable number of tandem effect (VNTR). The technique are used earlier involved southern blot hybridization using radio-labeled VNTR as a probe. It includes first isolation of DNA, second digestion of DNA by restriction endonuclease, third separation of DNA fragment by electrophoresis, fourth transferring of blotting of separating DNA fragments, synthetic membranes such as nitrocellulose or nylon, 5 hybridization using labeled VNTR probe, and 6 detection of hybridized DNA fragment by autoradiography. A schematic representation of DNA fingerprinting is shown in figure 5.16. The VNTR belongs to the class satellite DNA, refer to many satellite or small DNA sequences arranged tandemly many copy number. The copy number is varies from chromosome to chromosome in an individual. The number of repeats shows very high degree of polymorphism. As a result of size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiography given many bands of different size. These bands give characteristic pattern of an individual DNA. It differs from individual to individual in population except in case of monozygotic identical twin. The sensitivity of the technique has been increased by use of polymerase chain reaction PCR. You will study in about chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application of forensic science, it has much wider application, such as in determining population of genetic diversity. Currently, many different probes used to generate DNA fingerprinting. Here, symmetric representation of DNA fingerprinting. Few representative chromosomes have been shown contain different copy number of VNTR. For the sake of understanding the different color schemes have been used to trace the origin of each band in gel. The two alleles paternal and maternal of chromosomes also contain different copy number of VNTR. It is clear that binding pattern of DNA from crime scene match with individual of B and not with A. Parental chromosome, maternal chromosomes, chromosome number 7. DNA from individual B, this is DNA from individual A. Chromosome number 2, and this is chromosome number 16, this is chromosome number 16. Number of short tandem repeats 11, chromosome number 7, 2 and 16. The बी एंड नॉट विथ ए। यहाँ पे देखो, यहाँ पे सीन क्राइम सीन का है, ये दो सस्पेक्ट्स हैं, ठीक। देखो यहाँ पे इसका यहाँ पे एक मैच हो रहा है, एक यहाँ पे, एक यहाँ पे, एक यहाँ पे, एक यहाँ। सो दिस इज क्रिमिनल। नंबर ऑफ शॉर्ट रैंडम रिपीट्स। Summary. Nucleic acid are long polymer of nucleotides, while DNA stores genetic information. RNA mostly help in transfer and expression of information. Though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable in better genetic material. However, RNA is the first evolved in DNA was derived from RNA. The hallmark of double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pair with thymine through 2 hydrogen bond, and guanine with cytosine through 3 hydrogen bond. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by complementary hydrogen bonding. A sequence of DNA that codes for RNA may be simplistic term can be referred as gene. And during transcription, also one of the strands of DNA act as template to direct synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene splits. The coding sequence exons are interrupted by non-coding sequence introns. Introns are removed and exons are joined to produce functional RNA and by splicing. The messenger RNA contains the base sequence that are read in a combination of three to make triplet genetic code. To code for an amino acid, this genetic code is read again on principle of complementarity by tRNA that acts as adapter molecule. There are specific tRNA for every amino acid. The tRNA binds to a specific amino acid at one end and pair through hydrogen bonding with codes on mRNA through its anticodon. The site of translation of protein synthesize the ribosomes, which bind to mRNA and provide platform for joining amino acids. One of the basic mRNA acts as catalyst for peptide bond formation, which is an example of RNA enzyme ribozyme. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive process, these have be tightly regulated. Regulation of transcription is the primary step of regulation of gene expression in bacteria. More than one gene is arranged together then regulated in units called as lac operon. Lac operon is prototype of operon in bacteria which code for gene responsible for metabolism lactose. The operon is regulated by amount of lactose in medium where the bacterium is grown. Therefore, this regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project as a major project aims to sequence every base pair in human genome. This project has yielded much new information. Many new areas and avenues have opened up as a consequence of project. DNA fingerprinting is a technique to find out variation in individual of a population at DNA level. Its work on the principle of polymorphism in DNA sequence. It has immense application in the field of forensic science, genetic biodiversity and evolutionary biology. Amplified repeated DNA, okay. Suspect B and subsuspect A. B and T are match हो रहा है, ठीक. PNTR size varies from 0.1 to 20 kb. In PNTR, variable number of tandem repeats. Polymorphism in DNA sequence is the basis of genetic mapping of human genome. Further, the polymorphism are inheritable from parent to children. DNA fingerprinting is the basis of paternity testing. These are genetic DNA fingerprinting. DNA fingerprinting involves identifying difference in some specific region in DNA sequence called repetitive DNA. Because in these sequences, small stretch DNA is repeated many times. A bulk of DNA form major peak. The other small peak referred to as satellite DNA. Silent feature of human genome. The human genome contains 3,164.7 million base pair. Average genome contains 3,000 base. Total number of base being dystrophin 2.4 million base. Largest human genome 2.4 million base. Total number of estimated genes 30,000, much lower than previous estimated 80,000 to 1 lakh 40,000. Almost all 99 point is not exactly same. Function of 50% function unknown. Less than 2% genome code for protein. Chromosome 1 most gene 2,968, Y gene 231 gene. Scientists identify 1.4 million locations where single base DNA difference SNP single nucleotide polymorphism pronounced SNPs occur in human. Frederick Sanger gave DNA fingerprinting. No. Fragments of sequence using automated DNA sequence principle given Frederick Sanger. But Alan Jeffrey दिया था ना DNA fingerprinting. Alan Jeffrey कहाँ दिया है? तू ही है आशिकी, तू ही आवारगी, तू ही है जिंदगी, तू ही जान. Hepatitis elegans and non-pathogenic nematode or the Rosabella fruit fly. Goals of Human Genome Project. Identify 20,000-25,000 इतना ठीक है. Bioinformatics ठीक है. Evolution black operar pressure negative okay. And here is lactose and allolactose. Operon. Translation. Growing up polypeptide chain. Bind tRNA. Translation requires polymorphism amino acid to form polypeptide. The order of sequence amino acid defined by sequence of bases. Charging of tRNA amino acceleration. सिक्कल सेल एनीमिया. ग्लूटामेट के जगह पे क्या वोलिन आ गया. यूजी स्टार्ट कोडान प्लस मेथियोनिन को कोड कर रहा है. ठीक. यूएजी स्टॉप टर्मिनेटर कोडान. यूनिवर्सल ट्रिपल कोड फॉर फेनाइल एनानिन. कोड आर यूनिवर्सल but some exception present in mitochondria, mitochondria and protozoans. Marshall Hillman का cell free system protein synthesis finally had been code for this. Code to it deciphered. Several enzyme polynucleotide polyphosphorylase. ओके भाई. हर गोविंद खुराना प्रोवाइड किए थे क्या ये बताया. मतलब प्रूफ ट्रिपल होता है. मोर डाउटिंग द केमिकल मेथड डेवलप्ड बाय जॉर्ज गोमो. ठीक है. टू टेलिंग एडेनलाइंस 200 300. RNA polymerase transcribe rRNA 20S 80S 5.8 or RNA polymerase third responsible for transcription. tRNA 5S RNA sRNA small subnuclear RNA. ओके ठीक है. फिगर देख लो. ट्रांसक्रिप्शन यूकैरियोट. कैपिंग इंट्रॉन RNA स्प्लाइसिंग G triphosphate 3 prime. Okay bro. Very similar experiments using radioactive thymidine to detect Distribution synthesized DNA chromosomes were performed on Vicia fava beans by Taylor colleague in 1958. The experiment proved that DNA chromosomes also replicate semi-conservatively. Michelson and Stahl experiment. The Michelson experiment based on density. RNA transmission of genetic information. RNA is better, but for storage genetic information, DNA preferred. RNA genetic material, for example, in tobacco mosaic virus and QB bacteriophage. RNA क्या होता है? जेनेटिक मटेरियल होता है. ठीक है ब्रो, ठीक है. हर्सियन ने अनइक्वोकल प्रूफ दिया कि इसका कि डीएनए ही है जेनेटिक मटेरियल. चलो भाई ठीक. ट्रांसफॉर्मिंग प्रिंसिपल कौन दिया? एवरी मैकलियर्ड एंड मैकार्टिज. आरएनएज और डीएनएज वगैरह मिला के. ठीक. क्या भाई, क्या गुंडा बनेगा तू. ट्रांसफॉर्मिंग प्रिंसिपल 1928 में फ्रेडरिक ग्रिफिथ दिया. स्टोफेज निमोनिया लेके इंजेक्ट करके. एस वायलेंट है इसका. यस और आर दो स्ट्रेन. ठीक है ब्रो. यूक्रोमेटिन is said to be transcribing active chromosome where heterochromatin is inactive. ये important है. non-histochromatin protein in type of nucleus there is region of loosely packed strand, light strand on the eukaryotic. DNA AD structure of the 1869 में कौन बना? फ्रेडरिक मिशर ने. 1953 में वाटसन और क्रिक का एक्सेलर डिफ्रैक्शन वो आया. क्या? डबल हेलिक्स. और एक्सेलर डिफ्रैक्शन कौन किसका ये था? ये था किसका भाई? मॉरिस विल्किंसन रोजालिंड फ्रैंकलिन का एक्सपेरिमेंट. ठीक है. 5174 5386 न्यूक्लियोटाइड. बैक्टीरियोफेज लैम्डा का 48502 बेस पेयर. रिसर्च गोली है 4.6 × 10⁶ बेस पेयर. ह्यूमन डीएनए सिंगल हैप्लॉइड रहेगा तो इतना. डिप्लॉइड है तो 6.6 × 10⁹ बेस पेयर. ठीक है. ये रहा चैप्टर 5. कुछ सिर्फ रीडिंग की है और क्या. इसमें कंसेप्ट थोड़ी एक्सप्लेन हो रहा है, सिर्फ रीडिंग हो रहा है. यहाँ पर प्रॉब्लम देख कहाँ आ रहा है मेरे को. प्रॉब्लम आ रहा अपन को. हाँ, इसमें थोड़ा प्रॉब्लम आता है. इस यहाँ कंसेप्ट ठीक है. यहाँ का कंसेप्ट समझने की ज़रूरत है. उसके बाद ये कंसेप्ट थोड़ा समझने की ज़रूरत है. थोड़ा सा पता अपने को. और इसको थोड़ा समझने की ज़रूरत है. इसमें भी थोड़ा सा अपने को कन्फ्यूजन रहता है. और क्या. और ये इम्पोर्टेंट टॉपिक है. हाँ जॉर्ज गमो. ठीक है. इसमें ये लर्न कर लेना है बस. यूजी डबल फंक्शन मेथियोनिन को और एसिटेट कोडॉन को. ये सब फिनाइल एलानिन को कोड कर रहा है. ठीक है. सिकल सेल एनीमिया में क्या ग्लूटामेट को रिप्लेस हो जाता है किससे? वालिन से. ओके ठीक है. डॉक्टर इसमें थोड़ा मिस्टेक है, इसको कर लेना. इसको थोड़ा मोड़ा देखना है इस फिगर को.